Zach is giving me the look. No. All right, so there was one slide left in protein characterization. Have a look at it on your own. It's not too essential. So moving on to vitamin analysis. So it's a short and sweet lecture. We'll see what we can get done in the remaining 20 minutes. We used to have a vitamins lab, vitamin C lab, but we substituted this year with the reagents lab. So yeah, we're not doing a vitamin C lab, but we're going to talk about the method that we would have done during the lab. All right, so fun stuff. Let's define vitamin. What is a vitamin? Nathan.
It's like an organic molecule that's needed in diet because it can't be synthesized by the body.
Thank you. And it's needed in a small amount, in a very small amount. Okay. Why is it important to analyze vitamins? Yeah, wow, great. Who do I pick? I'm going to go with Claire.
To meet like nutritional values of particular food.
Exactly. That's what you're going to say? Daniel.
Well, I was just going to say for nutrition labeling.
Okay, so related, but okay. They can be toxic in high amount. That is very true. But I didn't have it on my slide. Yes, Sam?
I was going to say, during food processing, they might get destroyed. And so
Exactly. That's a big one. Determine the impact of processing on the retention of vitamins. For example, vitamin C is currently on the label. It will be removed once the new label is in effect. But vitamin C is one of the most prone to processing degradation. So it would be usually what they do, and that was part of the lab, is we make sure that producers put in vitamin C above and beyond their claim, because during processing and during shelf life, you would, during storage, you would lose some of the vitamin C. Okay. So here's the definition of the vitamin, and to assess diet adequacy, nutritional labeling, and determine the effect of processing. All right, so what are the two vitamins? One of them already said vitamin C. What's the other vitamin that is on the label? Yes, Zach?
are we talking about the new label or the
Old label.
old label? It's A and C.
Right, exactly. That's the old label. Currently, it's A and C. And sometimes, if a product is fortified, they choose to list other vitamins present. It's not mandatory, it's a choice. Okay, now the new label has only vitamin D. And since originally there was concern about deficiency of vitamin A and vitamin C, and this nowadays there is no longer concern about the deficiencies of these two vitamins given how many foods are there fortified with these vitamins. However, the concern currently is with vitamin D deficiency, and that's why we're moving into vitamin D on the nutrition label. So just refresher so that you know which ones are water soluble and which ones are fat soluble. Vitamin C and a bunch of vitamin Bs, B1, B2, B3, B5, etc. up to B12. These are all water soluble. And this is important to differentiate of which ones are water soluble and which one is fat soluble. Are fat soluble due to the way we prepare our samples for analyses. So those that are water soluble, the extraction usually is water-based, aqueous-based with some acid or alkaline and some heat to extract them. Whereas those that are fat soluble, the process involves, because they are associated with the fat component, so to release them from the fat component, usually we do saponification step. So to saponify, to solubilize the fat components, so these can be extracted with an organic solvent away from the lipid fraction. Okay. So this is just FYI, the year of discovery, their vitamin letter name and their actual names, and then the source. This is FYI for you. So the vitamin chemical structure is so diverse. It's not like all amino acids have similar structure, all sugars have similar backbones. It's not the same. Every vitamin is different, and some has, for example, nitrogen in them. Several of them has a nitrogen, and here, for example, vitamin B9, which is folic acid, has a glutamyl groups, glutamic acid group attached to it. Others have ring structure in them. Yeah, so they are very different. Because they are very different, is it possible to analyze all vitamins following one technique? No, it's not possible. Due to the diversity of these vitamins and the different solubility of these vitamins and different reactivity, so they won't be able to analyze them using one method. There are different categories of methods and different methods for each of these vitamins. So we have bioassays, which involves a rat model, in vivo assay. For example, there are standard methods for vitamin D and vitamin B12. Microbiological assays, these are assays that have based on the growth of a microorganism. So we measure the growth of the microorganism based on the concentration of a particular vitamin in the media. So these common ones are niacin and folate that can be analyzed using microbiological assays. Chemical assays, the rest of the vitamins can be analyzed by chemical assays, and usually measuring fluorescence or absorption, and oftentimes using like vitamin C is a titration method, and there's a fluorometric method, whereas vitamin A and vitamin E can be measured by HPLC, for example. Can be extracted, and an HPLC method can be used. So almost all vitamins are sensitive to something. So some of them are sensitive to pH. For example, vitamin C at their alkaline pH, they are, yes, Danny?
Just curious, since there's nitrogen in a lot of the vitamins, why don't they cause overestimation of protein analysis?
They do, and that's why we call protein as crude measurement of protein, because they are quantitated as nitrogen, and we overestimate the protein content, and that's why it's not an accurate measure of the protein. It's a crude estimation of the protein. But usually vitamins are present in really small amount, so their contribution is not that huge. Yeah, Sam, you had your hand up.
That's what I was going to ask, is like it's such small amounts.
Yeah, it's a very small amount. It's not really going to impact that much. Yes, sensitivity. So I was saying that pH, for example, vitamin C is sensitive to alkaline pH. So under alkaline pH, it's more prone to oxidation. Light, vitamin riboflavin, for example, and vitamin A are very prone to UV light, especially riboflavin. So if we're wanting to analyze riboflavin content, extract it and analyze it, usually it's done in under subdued light. So usually there are special rooms. If your company analyzes riboflavin very frequently, there is a designated lab space where the light in the room is always dim and there is no UV light. Oxygen, several of these are prone to oxidation: vitamin E, vitamin C, folate, even vitamin A. Sometimes in the analysis they use, while you're preparing your sample or extracting your vitamin, they put antioxidants in there, reducing agents. Beta-mercaptoethanol, for example, is very commonly used during the preparation for the analysis of these methods to avoid oxidation. Some are prone to temperature degradation. Vitamin C is one, vitamin A is another. Um, so again, avoid high temperature when we're preparing samples for analysis. In the past, when we used to measure vitamin C in orange juices, one of the samples were frozen concentrate. So the frozen concentrate, at one time we did not thaw it for the students ahead of time. What we did is we asked the students to thaw it under warm water. Yeah, no, that wasn't a good idea because the vitamin C was underestimated in these samples, and we deduced that the water was too warm, that it actually degraded the vitamin C and ruined the analysis. So if we are analyzing content in a complex matrix, obviously we need to extract them for analysis. So you have the water-soluble vitamins, which often we use heat as well as either acid hydrolysis or alkali hydrolysis to extract those vitamins. If we are using a fat, or if we're analyzing fat-soluble vitamins, we need to do saponification to break it from fat components, and then use an organic solvent to extract it, and yeah, and then they will be ready for analysis. Enzymes sometimes are used, and I'll talk about they're used for folate, for example, because folate or folic acid can be bound to proteins and starches, and also it's conjugated with the glutamic acid. So in order for it to be analyzed, they use a tri-enzyme kit where they put protease, alpha amylase, and conjugase to release that vitamin for analysis. Precautions, we already mentioned some of that. So antioxidants such as beta-mercaptoethanol, we call them reducing agents, are added during vitamin C and vitamin A and folate, because all of these vitamins are prone to oxidation. So during sample prep, we add antioxidants. Also during sample prep, if we're preparing our sample and we have a bunch of samples and we're not ready yet to analyze everything, we flush with nitrogen. That means we remove the oxygen and replace it with a nitrogen layer, and then we close our container until further analysis. So that means we're removing the oxygen to prevent oxidation. Chelating agent. This was actually something that I remember with the capstone group. They were measuring vitamin C in a product that they added fortified with vitamin C and also iron. And then they were trying to measure vitamin C, and they weren't able to detect any. I was like, but they got confused because they put a lot of vitamin C in there. And then they came to me and asked what's going on, and I said, what's in your sample? And they, by passing, they mentioned they had iron. Well, iron enhances oxidation. So in the presence of iron, and then there is your vitamin C and processing, heat, there you go, you destroy all your vitamin C for analysis, basically. It interferes with the analysis. So EDTA is usually added to chelate the iron in order for the vitamin C to be able to be detected and not get to an oxidized state. And I'm going to talk about different states of oxidation for vitamin C, but the initial state, or the primary state of oxidation, L-dehydro ascorbic acid, is an oxidized state of vitamin C, but it's biologically available. So although it's still biologically available, certain methods cannot detect it in this form. So the chelating agent prevent that from happening. The chelating agent prevent the iron from causing that primary oxidation of your vitamin C. pH, we control the pH. An example for analysis of vitamin C, we make sure that there is an acid in the sample prep so that the pH is low enough and not to induce oxidation. Subdued light for vitamin A and riboflavin, given that are both sensitive to UV radiation, and then also making sure to avoid heating. Example for folate and vitamin C. We'll hold off on the recap questions for now. So bioassays, that means we're using an animal model, and usually it is for the two vitamins, B12 and vitamin D. And they are official methods of analysis for certain foods. So basically we feed animals, usually rats, and then sacrifice them for measurement. Vitamin D, for example, the test is called line test because it's based on the calcification of bones. So a specific bone is pulled and the amount of calcification is measured on that bone. And it's directly related and proportional to the concentration of vitamin D. So the way they do it, they would bring a certain number of rats. There is a depletion phase. That means they feed them diet that is depleted of vitamin D, doesn't have any. And then they feed them for a known amount of time, for a certain amount of time. Standards, that means known amount of vitamin D, and unknown amounts of vitamin D. That means the food product you're measuring the amount of vitamin D in. And then they measure bone calcification, they do a standard curve and using this known amount of vitamin D, and then they detect the concentration of vitamin D in the unknown samples. Microbiological assays. So here, as I said earlier, here you measure the growth of organisms based on the amount of vitamin present. And the growth is measured by measuring turbidity. So when you have cells growing, turbidity will increase. Also, sometimes they're acid-producing bacteria. So if you measure acid, it's related to the growth of the bacteria. Or you can measure gravimetrically. That means you weigh the cells that have been produced, the microorganism cells, or measuring respiration. So there are different ways of measuring the growth of microorganism based on the concentrations of the vitamins you have. Niacin and folate can be determined using this technique. For folate specifically, they use Lactobacillus rhamnosus, and the tri-enzyme I mentioned earlier, alpha amylase, protease, to break down proteins and starch and release the vitamin, and conjugase to remove the glutamyl group, and then measure turbidity at 550 nanometer after a certain period of time. Chemical methods for fat-soluble vitamins. So saponification, like I said, to release it from the fat component, and then the vitamins are extracted with organic solvent, and then they can be measured by HPLC. Common HPLC is the normal phase, and then UV detection or fluorescence detection. All are AOAC methods for detection in different types of food. So this was the method that we did in the lab for vitamin C analysis, but we still have 2 minutes, but I don't want to start it and stop after 2 minutes. Doesn't make sense, so I will continue on Wednesday.